Well, I think I will start now. Thank you for coming, obviously. This presentation is something that, well, risk of bias assessment is something that I started to think of a long time ago when I did my first meta-analysis in 2000. It was published in 2015, so I'm not sure when I started. It took a long time. But later with time, somehow it always like dragged along all of my meta-analysis, this thinking of I don't trust every study the same, so how to capture that. And with that, we started to work on this issue of assessing risk of bias in, I would call it classical ecology and evolutionary biology, which is not the same as most of these like environmental evidence synthesis do. So it's kind of done by small teams of often of people who are not very well trained in meta-analysis or systematic reviews. And today we'll just have a really short kind of introduction by everyone because I don't think it's that many people. So I would just like to know what's your field of research, if you can talk. I don't know if that's an option. I guess it is in the Zoom. And what's your experience with conducting risk of bias assessment? After that, I will present our recent work. We just submitted it 2 days ago on the issue of risk of bias in ecological meta-analysis, where we propose some actions how to change the current situation, which is quite bad, a spoiler. So I would like to hear opinions on these actions that we suggest. After that, I want to show you 4 basic, as Alfredo says, yet powerful risk of bias questions we want to introduce as a simple start for ecological and evolutionary meta-analysis. And then there will be a practice bit after the break, where basically you will try to use these questions to answer those for some papers in ecology or evolutionary biology. That's going to be for half an hour. After that, I would like to spend some time in getting your feedback on these questions. Are we missing something? Is it too basic? And finally, because these are some basic questions, we would like to develop a more suitable tool or tools. And in that, many of us are not experienced, so we would like to get some, well, experiences, ideas, and similar, especially from people who have conducted these things or developed some tools. So to start with, can you just let me know what's your field of research and have you done any risk of bias assessment, if you know what the risk of bias assessment is? Somebody has to start, Losha.
I knew you're gonna ask me. Yeah, I have experience in meta-analysis. My background is ecology, evolutionary biology, ecotoxicology, and we do a simplified risk assessment, risk of bias assessments for our meta-analysis whenever possible. And sometimes it's generic, sometimes it's more specific. So yeah, you know what I'm doing. And we also developed some tools for assessing, but evidence synthesis, so meta-analysis and systematic review in EcoEvo, like Prisma EcoEvo.
Okay, yeah, thanks. Alfredo?
All right. So yeah, also from ecology and evolution, I have experience with meta-analysis. I must confess I've never done a proper risk of bias assessment. We've done a few attempts at assigning or categorizing studies based on a few traits such as selective reporting, blind data collection, and things like that, but never like a proper risk of bias assessment. I find it a little bit overwhelming and scary, so I would like to have some simplified thing that we can start using to get into motion. I think that might be the barrier for many people. And after that, maybe we can implement like complicated tools.
Yes, thanks. And Heikel, I don't know if you have option to tell us about yourself.
They've put in the chat. Sorry, I cannot unmute myself. I'm a PhD student in the field of ecology, and the subject of the workshop is new for me.
Okay, good. Then, well, let's start with my first meta-analysis, and that was about fitness consequences of divorce in monogamous birds. It was a very interesting experience that I think lasted through all of my PhD. So I started in the first year and I actually finished. That was my last paper that I finished because I did it over and over again. I didn't have anybody to really help me with that. So it was a lot of mistakes, but I think because of that, I also learned a lot, probably more than I would have learned if I had some guidance. One thing that obviously I found interesting is once when I read these papers that I wanted to include in meta-analysis, many things did not make sense. So values that were reported in, for example, a figure would not match the values reported in the text and similar. And that made me also, I often didn't trust the methods, so they were very unclear. So I did develop some really like my own, not very systematic, a tool to assign a trust that I have in each study on a 1 to 3 level. And actually, I put that as a moderator in meta-analysis, and it showed some moderate influence on the effect size. So basically, studies that I trusted less did show slightly higher effect sizes. Now looking backwards, I did not do it really properly, but I think it was also good just thinking about how this can influence final results of a meta-analysis. And how I see it is that ideally you have a lot of studies. These are ingredients that you use to make meta-analysis or systematic review, which is a nice cake ideally, but often people don't do meta-analysis very well. So you can potentially get good ingredients, but then with bad end result. However, and I think the evidence is piling up, that in many fields of research, including, unfortunately, ecology, we don't have a high-quality evidence. So then the question is, can you ever make a good cake out of bad ingredients? Because meta-analysis or systematic reviews should be as good as the individual studies that we include in meta-analysis. For example, and this is from one of our papers, where we looked into very broad set of papers across ecological subfields. A lot of work remains unpublished. Really, I mean, this is really shocking amount of work is badly designed. So almost 70% of primary studies in our sample. And many studies are underreported, so they don't fully report their results. For example, they say, we did not find any significant correlation between A and B, but that's all they say. So they put this P larger than 0.05, and then it ends there. So I think out of those ingredients, it's very unlikely that you will make a nice cake, but rather you will make this a bit messy cake. So at least we should acknowledge to what extent we do make a messy cake, because obviously some things we can do better. For example, methodological analysis approaches you do in meta-analysis, unbiased screening, and similar. But there are things we cannot change, and this is the evidence that gets into our evidence synthesis. So it is as it is. We just have to acknowledge somehow that some of this evidence is stronger, some of this evidence is weaker. And obviously, there is this question of what does it mean a good study? And I like this representation of different aspects of, well, study quality or goodness, because you can say definitely that studies that have high bias, so that means that they consistently show a certain type of evidence of effects that is not really true effect, and that have high variance are bad studies. Those that also have low variance but high bias are also probably bad studies, because they don't give you at all, they don't hit at all in this middle point of real true effect. And then obviously you have studies that are overall hitting somewhere in the middle, but around this middle, but without any bias. So this is considered still, I guess, good evidence, but slightly less able to point into exact effects. So you get, for example, larger confidence intervals. And then something that I don't think ever happens in any normal field of research is this low bias and low variance. I'm not sure if that, it would be interesting to see if there is an area where you really have low variance and low bias. And when I started to then kind of more systematically read about the risk of bias, it was also interesting to see that people in different fields, but also subfields, use many different terms, which is a bit confusing. So they use terms such as risk of bias, but then you have study quality, then you have critical appraisal, reporting quality, methodological quality. Is that the same as study quality? Or so it's very messy, actually. And then I try to structure it in some main biases that can happen at the level of meta-analysis. So one is observer bias. So you as systematic reviewer or meta-analyst are prone to biases. And that's why often we try to do meta-analysis in a team. So we try to reduce maybe these types of biases. For example, it's been shown that certain people are more likely to exclude evidence that does not fit their idea of what effects should look like and similar. But they find some explanation for it, but still they're more likely to exclude it. Then we have these study biases. So these are biases of the studies that we include in meta-analysis. And finally, we have biases at the level of the overall evidence you have gathered. And this is, for example, publication bias. So studies are just those that have been published. Language bias, because you're more likely to find studies in English. Then taxonomic biases. So research has been just done on a certain amount of species, and often these are those that are easier to study and similar. And what we are interested when it comes to risk of bias assessment is these within study biases. They're opposite to the internal validity of the study, which refers to the extent to which a study that you conducted can establish a causal relationship between independent and dependent variables free from confounding factors. And it seems maybe kind of like, okay, we can do that, but then in reality, it's very difficult to actually do this properly. Not only because researchers are not trained in how to do things well, but also because, especially in ecology and observational studies, there are often things that you just cannot remove. It is as it is, and you cannot change it. So the question is whether your study design and execution, whether departures from some ideal study could equally explain the finding. But then other terms that are related to within study bias are methodological quality, because often within study bias is assessed within a broader area of methodological quality of the study. And reporting quality, because if something is not reported, you cannot assess it. For example, blinding is a very common example in ecology, where studies commonly don't say we blinded ourselves to collecting data. So likely they did not blind themselves, but it's not explicitly stated in the study. So you can't assess within study bias without proper reporting in the study. And then all of these together are often referred to as parts of critical appraisal. But the thing is that critical appraisal can also include just within study biases. It can include within study biases plus methodological quality plus reporting quality. So it really depends on the tool itself. So for me, when I started to, as I said, systematically kind of explore this area, it was a bit confusing and overwhelming to read all of these different papers from different fields and try to make sense out of that. In some fields, such as medicine, there are many different types of risk of bias assessment tools. So basically these are, let's call them list of different questions that try to help you to understand the level of bias. So does it exist actually, or it does not within primary studies? One important thing to say here is that what I said just now, level of bias, you cannot assess that. So risk of bias studies just say there is high risk of bias usually or low risk of bias. There is no really saying how much this affects the result. It just says it's present, so there is some risk of bias, but it doesn't mean that study results are biased, obviously. It can still happen that they are not. In environmental evidence, collaboration for environmental evidence, there is a CSAT tool, I don't know how to say that in English. I tried to use it, and I have to say it was so difficult. I just didn't understand many questions, and I gave up on it. And I have some colleagues that have tried. They have also gave up on it. But again, we are just ecologists. Evolutionary biologists is not really this like environmental studies that usually have some, let's say, more precise designs. So what I just said, a few important notes to consider is that this risk of bias assessment usually aims to evaluate how susceptible primary studies are to biases, but we don't aim to quantify exactly, is it like 1 or 2 or 3 or 10, for example. And then once you assess this risk of bias of all the studies that you include in meta-analysis, you try to then put results or interpret results of your meta-analysis in light of these biases. And obviously, there are different options what you can do after you score the studies. If you ever went to, for example, Cochrane review database, and I do sometimes when there's something medical and I'm not sure what I should decide on, and you look at the evidence for a certain, let's say, treatment, is it effective or not, many of these reviews tell you this is the overall result, but the underlying evidence is not of high quality. So they don't remove these studies. They include them. They do meta-analysis using all of these studies, but they are cautious and they just say, you know, this is what we can find out of the studies, but we don't trust these studies a lot. And that's medicine and that's human health, where studies, you know, I always think should be of really high quality. They should be in ecology too, but I guess there is a bit less push because it's not influencing human well-being directly. So what I mentioned earlier on is that when you try to assess risk of bias, if you ever did try, and a few of us have, the issue is the low quality of reporting in primary studies. Sometimes it's difficult to say because you're not such an expert if that particular type of method. Because what you often get in ecology, you're expert in a certain field, but sometimes some studies use some approaches that you just don't know, like you cannot say how they actually work. So it's difficult to judge it. And if you use any of these already developed risk of bias tools, it takes a lot of time to assess risk of bias. I never done it, but that was my estimate, so I gave up ahead of trying to assess something. And recently there has been this paper, and they looked into how much meta or systematic reviews in ecology actually do include risk of bias assessment. So here is on the x-axis is the number of systematic reviews, and there you have the years in which these were published. So these darker bars are those reviews that do have some kind of risk of bias or critical appraisal. So as you can see, it's extremely low. I think it's around 2% or something overall. So that's almost nothing. In medicine, I think Alfredo showed that yesterday they have around more than 90% of reviews would have risk of bias assessment. So we were interested in why researchers don't use risk of bias assessment in ecology. So we wanted to know how familiar are they actually with the risk of bias assessment. And we conducted a survey that received 232 responses. And we were also interested in whether journals have some guidelines on authors of meta-analysis that say you should conduct some kind of quality appraisal of the studies included. And results are as we expected. So half of our respondents have not ever heard of risk of bias, and then half of them said that they have heard of it. But when we looked into their responses, only actually 28 had a correct understanding of what risk of bias is. And almost half of them thought that risk of bias is a publication bias. and it's not. So risk of bias is at the level of primary study, and publication bias is at the level of the overall pool of studies. They also do not conduct risk of bias. So of those that said that they did hear about risk of bias, half of them have not conducted, claimed not to conduct it. 53 said we have conducted it, but again, most of these actually conducted a publication bias analysis. And only 10 people, so this is 10 out of 232, have actually likely conducted risk of bias assessment in their meta-analysis. And what they said, we asked them to rate their experience. Most of them, so 7 of them said it was very difficult. Yeah, obviously these existing tools are difficult to use. Then we looked into journals. So we asked whether a journal mentions that they, do they explicitly state that they publish either reviews, which is a bit vague, obviously, or evidence synthesis. And 58 do. For 151, we were not sure. So they just say, we publish reviews, but it's unclear what the review means. Is it a narrative review? Is it a systematic review? So we still kept those. Those that said that they didn't publish this type of articles, we just excluded. So then we looked, do they mention any guidelines that are risk of bias guidelines or that mention risk of bias guidelines? For example, reporting standards such as PRISMA, in there you have one question that says, has risk of bias, or I can't remember, critical appraisal being conducted? So we looked, are these ever mentioned? So most of them actually do not mention it, and those they do, don't mention it often in reference to study conduct, so meta-analysis conduct. They mention it in reporting guidelines. So they just say, follow ROSES or follow PRISMA. And when you open PRISMA, of course, it states there's some element of risk of bias. But only 6 explicitly stated, we want you to consider risk of bias or critical appraisal. And those that, oh, I missed one box here, those that are where they just publish reviews or similar, none of them explicitly links to risk of bias assessment as a part of conducting evidence synthesis. So obviously, there is a lot of room for improvement. So our first suggestion is to increase the awareness of researchers that this is extremely important element of systematic reviews and meta-analysis. But second, to provide guidelines on how to do it, and that journals should put some effort in having such guidelines as a part of their author instructions or similar. Then the second thing was to obviously facilitate risk of bias assessment, because currently it is difficult and some people probably don't do it because it is difficult. And the third part is to make it possible. So you have to have reporting standards that are as such that you can assess risk of bias in primary studies. So this is just what I said. This is very important, interesting paper where they assessed how long it takes per study in medicine. It's a clinical randomized trials. How long does it take to estimate risk of bias? And it's a long time. So it's 358 minutes per study. An average meta-analysis in ecology has around 40 studies included, so that would mean around 240 additional hours of work per meta-analysis. And it will probably be longer because people are not trained in it, so it would probably take longer than for medical people who often do these meta-analysis in these large centers such as Cochrane, compared to ecology where people are just not trained in it. So now I would like to have some general feedback on what I have presented now. So do you have any thoughts? Are you shocked? Are you not surprised at all? And I would like to hear about what you think about these proposed measures, especially the first one, where we would like to raise awareness and provide guidelines. Because the thing is, when all the meta-analysis courses that I've taken, well, this was back in the past, none of them ever mentioned risk of bias assessment. I did not take medical ones, so it was more ecology and stuff like that. But it's rarely mentioned. Sometimes they say, oh, you should also think about quality of the primary work, but nobody's really taking this, in my opinion, very seriously. So yeah, I would just like to have a short discussion on this. And here in the chat, I will put a document, Google Docs that I made for this workshop. And you have discussion on suggested actions section, where you can either yourself add your thoughts, or I can write them if you say them. So what I currently put in there is that obviously risk of bias has to be included in training materials and workshops. Journals and funders should provide clear reference to risk of bias assessment and invite more articles that would discuss this topic. So it like either, the issue of the journal about research quality of meta-analysis and similar. So I would like to hear if anybody else has any additional thoughts on this. What would help you to start using risk of bias assessment? Anybody?
I guess I can start.
Thank you. Okay, good.
That was really interesting. Absolutely fascinating to see how little critical appraisal and risk of bias is considered.
Yes.
I work mostly in evidence in environmental management, so slightly different topic. And we have found really similar levels and not reporting and stuff like that. So interesting to see that it's continuous through. When you were looking at the journals, and you said that they say make sure it's included in the reporting, what were you looking for in particular that was missing? Is that, so I have contacts with journals and I might be able to like bring this kind of thing to them as well.
So basically when you go to a journal guidelines, and first they don't have specific, I think few journals actually had meta-analysis type of article guidelines specifically.
Right.
Many of them just say we publish evidence synthesis and that's it. And then you see reporting standards, general reporting standards, either of publisher or a journal. And then you have, if you're doing a systematic review or meta-analysis, you might consider reporting standards such as ROSES, PRISMA, and similar. So it's really about reporting, not about the risk of bias itself. Only 6 journals did say something along the lines, you need to consider the quality of primary evidence, and then they link to, for example, that was the Environmental Evidence Journal and a few other ones that specifically say this is a standard of conducting this type of article. If you want to have it published, it has to consider the element of risk of bias. So I find these that refer to reporting guidelines really indirect. I mean, you can just skip it because it's reporting basically.
Yeah.
So I think it's extremely low a number of journals.
Yeah, I think that you're right, and I think probably that editors and like journals just aren't aware that this is one of the things that we're looking for. So I think that that's a really good step. If we can get the editor teams on board with incorporating this, I think that would be really powerful.
Yeah, and what we noticed, and I think it links to the issue, and this is that, as you could see, often they don't understand that there is difference between review and systematic review, and so they just say we publish reviews. So what are these reviews? So it seems that they don't see it as a really standalone type of work. So it's not the same as primary article, it's something else. And so if they don't see it in that way, it's also unlikely that they will think about, well, this type of study actually does need some different reporting standards, that does need some reference to how to conduct it and similar. So it just seems like elevating the importance or the distinction between meta-analysis, systematic reviews, and other types of articles, perspectives or research articles and similar. I think one part of the problem is that. Do you have any experience in getting like journals on board and stuff like that?
Yeah, so our center, so I'm part of the Canadian Centre for Evidence-Informed Management. We actually have an agreement with the Canadian Science Publishing Group, where we've embedded some of our employees as consultants. So if they are looking to incorporate evidence synthesis into one of their newer journals, they come to us with questions about how that they can do that. So having those kinds of relationships might be really useful. And we've also had experience, just like my professor and PI, he does talks sometimes for publishers just to explain why there are differences. So maybe having those kinds of discussions in ecology might be helpful as well.
Yeah, maybe something we have this Society for Open, Transparent, and Reliable Ecology Evolution. Maybe that's something that can also be a part of the, well, advocacy for changing the standards. It's not really about the openness, but it is part of this, like getting science to a higher bar of quality. Thank you, Megan. Anybody else would like to share some ideas or thoughts? Ryan?
Yes, just a quick one. I don't know if you're aware of any other ones, but I know the Latitudes Network has a list of risk of bias tools. So sites like this that list the tools and checklists you can use are quite useful because it's for different disciplines altogether. I'll put the link in the chat.
Okay, thanks a lot. I haven't heard of it. We found some papers that list different tools, so they looked into what tools have been used in certain fields. So we did made some internal list, but yeah, this is great. Thanks. Anyone else, or we go to the next part of the workshop?
Maybe just one brief
Yes.
comment. I was wondering, so I was thinking like, how can we promote risk of bias as reviewers or editors? And then my immediate thought that would be that we could ask authors that submit systematic reviews and meta-analysis to do it. But then I thought, well, I mean, that's a little bit unfair to ask for that if nobody is doing it. But maybe even if we should not ask to do it, we could ask to report they haven't done it as reviewers and editors, at least to get the conversation started. Maybe we could even have a couple of sentences, standard sentences that we write in our reviews and editorials where we say to the authors, like, I don't know if you're aware of this. This is very important. It's true that it's not yet ready, but we should be doing it. I understand you might not want to do it, but we should start reporting that we haven't done it so that we are aware of it. Some idea like that we could write like 3 sentences.
Because you always feel like you can't do anything to the system, but as a reviewer, you can actually have this feedback, and the editor would read it because they have to read your review. So, with time, it might get into their head that this is something that should be included as a part of meta-analysis. But again, there is this issue because, with this classical, if you heard about Brian and Noshek pyramid of culture change, at the bottom, you need to have infrastructure and help. And only then you can impose things. So one thing that we definitely don't want to for it to happen is that the journals say you have to do risk of bias assessment, while we don't have good tools for ecology, and we are going to put so much strain onto researchers doing meta-analysis. So I think that there needs to be some kind of parallel development.
A second thought is that I think we may have more meta-analysis doing risk of bias than we think we do, simply because people might be still trying to categorize certain qualities of the studies and testing for it with meta-regression and not knowing what they are doing is called risk of bias. I don't know, comparing experimental design or even observational versus experimental studies could be a question of risk of bias there. So, and thing is simply a lot of it is an awareness.
Yes, good points. Okay. They're partly taken because I'm not good at listening and thinking and writing it down at the same time. So feel free to just enter it yourself if I don't manage to capture it. Okay. Then I would like to present, and I would really love people from environmental evidence to then have a go at these questions that we are proposing. Because we were thinking about about it a lot. We started with a list of 7 questions, then realized that some of them can actually be merged for the simplicity. And this is what we came with. So I want to just for you to understand our starting basic points. And first one is that we wanted to have at least some basics covered because nobody is using it. So I understand that ideally you should start doing something straight away as it should have been done. But I don't think this is something that we can achieve in ecology at this point. So it has to be something that people might actually use because it is simple and it doesn't take too much time. Second, it has to be applicable to really broad range of evo studies, and really in ecology and evolution you have so many different types of studies. We also wanted it to make it quick to use, and we are aware that they do mix otherwise often separate aspects of the risk of bias. But I said our reason to do this was to make it as simple as possible. Ram, did you raise your hand afterwards, or was it?
Yeah, just to let you know, you're on presenter mode again.
Oh, okay. Oh, God. This is my second screen. Okay, but we'll have a break soon, so we can just stay as it is. Nobody minds. So our first question is, was randomization applied? So obviously in experimental studies, randomization is that you assign experimental and control units randomly to treatments or units, whatever they are they individuals or plots or whatever. So for experiments is relatively straightforward. For observational studies, it's a bit difficult because often you can't choose. It's just like how it is in nature. Plots are where they are. You sample from where you can sample. So here, causal inference depends on design or statistical strategies to approximate random design. So it can very rarely be really truly random. So the method of sampling from the population is normally considered separately as a factor that influences generalizability and not as a part of randomization. Another aspect of this is also, for example, capture marker capture studies. So you often here selectively have animals that are more likely to be captured. And in classical risk of bias tool, this would be considered under selection bias. But we clump it all in this randomization part of the question. Second question is, was confounding considered? This is only relevant, obviously, if you could not have a random design. And if the answer to the first question was no, then you might be interested in whether some uncontrolled or inappropriately controlled variable that influences both the intervention and exposure and the outcome is present in your data. But the thing is, it's very difficult to control for potential confounders in observational studies. So how are you going to control it? You can control it in your analysis. But then you come to another issue that in ecology, when you do meta-analysis, you do it on different habitats, often on different species. So it's across taxonomies. And that means that confounders might be different between these in reality, but also how do then clump together these results of some kind of analysis that have controlled for your confounder, but then these results are not comparable. So it's kind of very difficult, tricky thing, I think. And yeah, we discussed this a lot within our group, and we did not come up with some good solution to how to really consider confounding or how to evaluate if it was really considered in a study, if it's observational study. And then 2 questions that are relatively simple, but these are those that, especially the fourth one, that you can never explicitly say. The first one is about whether methods for measuring the outcome data were the same across groups or individuals. For example, you have one researcher always sampling from one population, and the other researcher always sampling from the other population, and they did not, for example, receive the same training treatment. So this might not be explicitly reported in the study. And then the fourth question was about blinding. And unfortunately, we know that studies in ecology never report on this. So we don't know whether they were blinded or not, but we can assume if they did blind themselves to data collection, they would say it. So if they don't say, they likely did not blind themselves to data collection. So these were our four questions that we thought, okay, probably people could be able to answer at least some of these. So what I would like to do next, before we have discussion on it, I would like to spend around 30 minutes on a little hackathon where you can choose whatever study you want. It can even be your own if you wish, within ecology and evolutionary biology. But ideally, it would be something that you are familiar with, so don't choose some topic that you don't know anything about. And then you try to answer these four questions with either low risk of bias, high risk of bias, unclear risk of bias. NA means it's not applicable. And finally, like, I just don't know, I cannot assess this. So it doesn't say it's low, high, it's even not unclear, it's just that you don't feel that you're placed to assess or answer this question. Then I would like you to note any issues you had. For example, I could not assess it because it's methodology I don't understand, or I don't understand the question that you asked. Any positive comments? This was easy. And at the end of the exercise for each paper you do, and I think you probably managed to do one paper, you score your experience from 1, which means it was easy to answer these 4 questions, to 10, it was difficult to answer these 4 questions. And you will do this via, I'll just give it to you now. I created the Google Forms. So you just, it's easier for you to do it in a structured way. You can find it in these Google Docs that I have sent, but I will also send a link to you in the chat. So the idea was to have a little break first, like 10 minutes, so your brains refresh, and then whenever you're ready, you can start working on one paper, I guess, is going to probably be enough. So let's have a 10 minutes break. Then I'll come back, and if you have any questions during your extraction, let me know. I'll be here on the screen.
Maybe one question.
Coffee. Yes.
So can we really use that in our own studies? Because that might be...
I know, but I thought about that, and I thought maybe it's like, in sense of you learn something, maybe you did not think about all of these elements when you were designing it. And also maybe it's going to speed it up because then you already know what you've done. But I understand it can be subjective. I don't mind. This is more of an exercise. So I really don't mind if you think it's easier for you to use something that you've done. So if I would do this for some meta study, then obviously I would not give people their own studies to assess. But I think for this exercise, it can be. And
I'll go for mine. It might make it easier to realize
yeah,
all these
exactly.
issues.
So try to be unbiased, obviously, in your head. If it's your study, it doesn't mean it's well done. I mean, I have many badly conducted studies in the past, so it's okay. I just wanted this to be as easy on you as possible and to see if these questions are something you can actually answer, maybe even for your own studies. And maybe in future we conduct a more proper type of hackathon using these questions. Okay, I'll go for a coffee break and I'll be back in 10 minutes. And you can start whenever you want on your questions. Okay. I'm back at the computer, so I would just like to highlight that in the Google Docs, I'll put it in the chat again. For each of these questions, you have some like more, I elaborated on each of them. So if you don't understand the question, maybe reading the full text can help you with understanding the question. Maybe it won't, but this is something that I would also like to hear from you. Like, I just don't know, like, I don't understand the concept, I cannot decide, and similar. Because I find it difficult myself, to be honest. I tried. It's not so straightforward somehow. And Alfredo, probably for you too, but working with these people that are more on the statistical side of things, I learned a lot through their comments on the manuscript when we were writing it. Like these were things I just really didn't know, and they relate to all of these different questions and what does it actually mean in practice. I see that somebody has just joined. That's Jacek. I don't know if you have any questions, because currently we are doing some hackathon type of exercise. So if you would like to do it too, let me know and I'll quickly explain you what has to be done. How is it going? Roughly. Okay. Hope others are good too. Okay, good. Is it are you still on the first paper, or you managed to go to the second one, the first?
I'm doing my own papers and it's hard enough, but I'm on my third.
Okay. I just didn't put, now I'm thinking about, if you're listening to me, maybe there the final comments, you can just add how long it took you to do it. So you have just some rough idea of how long does it take. I should have put it as a separate question, but I haven't thought of it before. So just add it as a comment at the end of the exercise. just a note, we have 5 more minutes. And I would not go over that time, so we can discuss this a bit. Is anybody done? Okay. I've seen some people nodding, but I cannot see people without the cameras on. So speak up if you want a few more minutes. If not, then I'll continue. Okay. Then we will continue. So, copy link. I'll copy the link to the Google Docs again. Okay, I see, Jacek. So sorry that you came in later because we were just trying out, Jacek, some 4 main risk of bias assessment questions that we propose as a starting point for ecology and evolutionary biology evidence synthesists to try to include in their evidence synthesis. So now I would like to hear your feedback on these questions. Obviously, I have it in the Google Forms, but still I would like to, for like 10, 15 max minutes, to hear your thoughts on how easy what this was. Is something missing? Do you think there should be more clear examples of what this exactly means and similar? Is it lacking? Should we have more questions that are more precise? Anything, basically any feedback you have on the exercise. Who wants to start? Losha? Also, I just want to point out that in the Google Docs at the top, you have this navigation bar. So if you click on discussion of questions, it will lead you to that relevant section of the Google Docs where you can write your ideas or comments if you don't want to say it out loud. So Losha, sorry.
Yeah, I'm facing the classical problem of reporting. Things are just not reported, and if they are not reported, we can make assumptions that they haven't done it, or that they've done it but just did not report. So they are, based on that, you can select like high risk or unclear, or I feel I cannot judge this because it's just not reported, so we don't know what happened. So we can go for, best scenario, like the most negative scenario, they haven't done it, but maybe they've done it. So to be fair, we should, for all my questions was, I cannot judge it. Okay, and I think we just need to, like, the easiest thing to do is just ask the question whether something was reported. And that's most fair assumption. Like, did they mention randomization? Not did they do it, but did they mention? Because this is the only thing that we can really tell from the report.
So what I've seen, it's commonly advised is basically if it's not reported, you call it unclear. And then this unclear is somehow the same weight as high risk of bias. So I put this question, I can't judge. And this was more about you don't have the expertise to judge, for example, if they are randomized. But what would you normally answer here would be unclear. So it's not reported, I cannot say because it's just not there. I could judge it potentially. And this unclear puts the study as high risk of bias. Yes, but many will be unclear. I think that's the interesting point.
I think 99% would be unclear because so that's one problem. And the second problem is actually, you can answer in multiple ways depending on how you kind of approach it. Like randomization could be done at different levels. So they could have done some, but not the others, and then it's kind of partial. Same for confounding. They could have considered one confounder, but maybe about 2 other confounders, but they didn't mention. So again, this would be partial, which we don't know, don't want probably. And same for methods of measuring the outcomes. Again, if they reported it, they could have done standardized measurement of something, but not fulfill this criteria for other outcomes, because usually there is more than one outcome.
Yeah.
So again, it would be partial because depending on the outcome and how likely to be biased, because some things are easy to measure, other things are difficult and more likely to be affected by, let's say, change of personnel. And there is no information about it usually anyway.
So I think you raised one important point that I have not highlighted, because this is assessed per effect size, I mean, per effect, not for the full study, because a study might involve different things, and not accounting for something in the design, for example, might affect outcome A, but not the outcome B. So I should have maybe highlighted that you choose like the main whatever analysis or something, but yes, it's tricky. After we discussed this amongst the group of people I worked with, we realized that because many will be scored unclear, it will also like show the message that we are doing it wrong. You know, if you can't score anything, that means that we just have such a bad reporting that it cannot be. So even if we can't judge it, I think it still will emphasize, if all meta-analyses do this and they say we can't judge, that means that we just have badly reported primary literature and it has to change. Somebody else, please? Any other comments? It can be also from you. Ryan, please raise the hand because otherwise it's difficult.
I think Alfredo was first.
Okay.
No, go ahead. I just about to raise my hand, believe it or not.
As just a comment about how far, I know you said primary studies, but should we be looking at appendices and like protocols on top of that?
I would say yes, because some information might be available there that's not in the main study. I mean, you often get that. More details on the procedure can be found in supplementary materials. So I would say, yeah, have a look at everything, but probably that should also be highlighted. So I'll put a mark to myself.
I had a thought about this unclear, and I have the feeling that there would be many unclears when you are dealing with observational studies than with experimental studies. Because I think experimentally, normally, let's say if they randomly chose the individuals at least or something like that. So I wonder whether we are conflating those two generally, and whether maybe one question would be whether the studies that you are meta-analyzing are experimental versus observational generally. That could be one of the questions for those meta-analyses that combine both.
Yeah, but I guess this doesn't go into risk of bias evaluation, but this is like a separate thing. So if it's observational, it's just more difficult
But didn't we say that the risk of bias pertains whether we can really establish a causal relationship between the two variables? And I mean, we can never do that, or almost never do that with observational studies. So being experimental already puts a step ahead at least.
Yes. But then you might like run into this issue of it implies that observational studies are less good, and they are not. They're just different, and by default they cannot account for the same things as experimental studies. And observational studies are extremely important. I mean, without them, I don't think you have any ecological relevance of whatever you observed in the experimental study. Well, I mean, you can, but often you don't. So I always am worried about like, then you see what I mean? It's somehow
Yeah, but we really have causal.
Imply.
We really have this word causal when we are defining the risk of bias. So it is implicitly saying that we are mostly interested in testing hypotheses rather than generating hypotheses. So I don't know.
Yeah, I mean, it's a good point. These are all things that I guess in the next steps of whatever we decide to do next, these things have to be discussed. Yeah, I mean, I always control in meta-analysis whether effect size came from experimental or from observational study. Another thing that I thought of, because I did try to evaluate one study, and they do say be randomized, but then when you read it carefully, they actually don't randomize properly. So that's another thing that you can run into. You see it mentioned, but actually it's it's not properly done, but because it's explicitly written, you just don't go further and you don't put extra time into really evaluating the method itself. Okay. Anybody else? Shreya?
So to add what is already said, but also I did like two different ones right now. One was a field experiment and the other was a lab experiment. And in the field experiment, they actually explicitly say we randomly selected the individuals to go into treatment or control. But in the lab one, they don't say anything explicitly, but I would assume so. Maybe they did it, or I can't make that assumption. So now it's not that only experiments, like if there are experiments, maybe they state it, maybe they don't state it. But my gut feeling would say in the lab, they must have, or they must have thought about something. But in the field, even though they say they did it randomly, they actually just go around looking for these individuals and then assign it to either treatment or control. But that could mean that there are other confoundings, that they went in a certain location first and then the other location and so on. So
Yeah. I mean, the thing is, this was a trick. Like all experience of working in this and thinking about this risk of bias made me also be desperate because you just run in circles. It's very, yeah, it's tricky thing. Like at one point I was just like, let's give up and don't dig into this because it's just so difficult and complex. And it's not just about risk of bias. It's about many different other things that can or cannot be accounted for. Yes.
just to add another thing, that I have done this assessment for the meta-analyses from which I picked this field experiment study. And I assessed 3 questions. One was whether they randomly like allocated the treatment and control, whether they considered some form of blinding. And the third was whether they had reported completely or not. And I think the randomization was rather 50-50 kind of a situation where 50% said they did and the 50 was unclear or no. And even though the ones that said yes, they did randomly, I could still again go ahead and categorize and say some of them are truly randomly assigning it and the others I can't judge it for. So just as an information that I tried doing these 3 questions for about 20-25 studies already in this.
Thank you. So what's the experience of the others? Did you answer to most of the questions unsure or I cannot judge? I just wonder, is this, as Loja said, the common experience in this group?
So that's what I said. For the one for
Yeah, for you.
missing, randomly is 50-50. That 50, I can say, yes, they are randomly assigned. At least the authors claim they have randomly assigned it. The 50 is like, no, it's unclear or a no. So I categorized it together because I can't say which one is which. And for blinding, it was almost largely unclear, or a few said why they could not perform blinding in these field experiments. And the missing data, of course, is like a rather, well, of course, but for this situation was a little bit easier for me to categorize. But then it was like 70-30 kind of a distribution. But I could do it in principle. It's just at what level of resolution I tried to do or couldn't do it. So that is it.
Anybody else wants to add something? Okay. Well, we'll see what we'll do after this. What are the next steps? How to do it? And are these questions good enough? I mean, it's going to hopefully be published at one point as it is now, or maybe slightly changed. But it's also, that's why we, in this paper that we submitted, we do not say you have to do this or what to do after you score the studies. Like, do you exclude studies with high risk of bias, which means almost all of the observational studies? That's not correct thing to do. So it's basically, yeah, we want to start conversation about introducing this and what are the pros and cons of introducing something like this in ecology and evolution, where many studies are observational, like 80%, I guess, of at least in my field, it's mostly observational studies. Okay, then, if no more comments, obviously you can add your comments after the hackathon, after the workshop or whatever this is, to the Google Docs. You have the link. And now I'm just going to go into the final part of this workshop. And that's actually a question that really interests me, and I don't have a clear opinion on it. And this is about, maybe, given what we've seen, do we need another tool? Because it seems that current tools are not used. Obviously, that can be caused by the fact that people don't know about risk of bias. So most of the people have never heard of it based on our survey, and they have a wrong interpretation of what risk of bias is. So obviously it can be because of that, but it can partly be because they went online, downloaded the tool, and then just said, well, this is not something that I can handle within my systematic reviews. So my main questions here is, do we need yet another tool? And how do we know do we need another tool? So how to know that we actually do need another tool? Then any tips and any experiences from people who have been developing tools? How do you go around about it? And how costly it is? Because we are thinking about maybe applying for some money to actually do this properly, but we even don't have the idea of how much work it is, how many people you need to work on something like this. And what we read from some guidelines on how to develop tools, obviously you need a list of experts who come up with a list of domains for risk of bias assessment. Then this goes into some kind of Delphi type process where every question is critically evaluated. Does it have a clear meaning? Does it make a separate explicit domain of bias and similar? And after you have some prototype, then you have to evaluate it. So here it is very important that there is high inter-rater consistency. So that means that it's very clear, each component of bias is clear, well explained, and thus everybody has the same or roughly same interpretation of it. Also, is it easy to use, and so forth and so on. And obviously all of these tools are developing along the way, so you have different versions of the tool. But yeah, I guess the main question is, do we need another tool, and how do we know that we do need another tool? So I would really like to get your thoughts on this. And also that's one comment section in the Google Docs, so you can enter your comments there too. So what's the future of this? So basically this is what we currently know. Risk of bias is never assessed. It's likely because people don't know about it, plus it's very difficult and complicated to do in small teams and not very trained teams in ecology and evolution. So what do we do next? Do we stay at this, or we try to do something, and what should we then do? Any thoughts? Nobody has any considerations, like
Well, maybe I can get it started so that others chip in. But I mean, I think, I like the experience from other fields, but I have the feeling that the type of studies, just thinking of experiments that we do in ecology and evolution, which is already quite a large field, are way too different from each other for us to generate a tool that would be applicable to any meaningful proportion of them. So I think that might be a strong difficulty unless you develop a tool for some very specific context experiments that people perform routinely and that they are published, like there is enough amount of scientific articles published on them. It might be not worth developing a tool and just focusing on making a few very easy questions and a few very easy suggestions of questions that people could consider if they can, and just implementing the effort in just trying to convince people to at least do those. But yeah, I don't know. That thing started. I guess somebody with experience on this, having used ROP tools, would be better suited.
Yeah. But I agree. I'm not sure if we need something because it's going to be very difficult to make it specific. Shreya, I think you wanted to say something. And then Washa.
Yeah, so I was also thinking maybe if it's not a tool, a guideline for how you can approach this would still be very useful. So if, for example, in a certain specific meta-analysis, I want to assess some questions, and I think once we kind of like go through the process of data extraction, we get some hint of what could be good things to assess. Maybe then having these guidelines with it would be helpful to kind of leave every specific field to be able to assess risk of bias on their own and develop something. Just a
That's
suggestion.
awesome. Yeah, I mean, I find it reasonable suggestion. Losha?
Yeah, I just wanted to go back to reporting transparency. I think we need to tackle that first before we can do risk of bias assessment. And even just having a simple generic tool for reporting transparency plus few other things. So for example, like what we normally do is whether they share the data and code, and also whether they report conflict of interest, because for some topics that's important, and funding sources, because again, if it's eco-evo, but it's relevant to industry or other policy, conflict of interest or funding could influence the findings basically reported. So those are things that are very easy to assess.
Yeah.
And they are kind of universal, more or less. And then, yeah, kind of assessment. I don't know, but the next things would be really the transparency of the methods. Do they feel complete enough to be reused or used in a replication study? So do they report enough the details to replicate what they did? Potentially, like theoretically, yes.
I mean, now ideas developing in my head, and that's about going actually larger than the risk of bias, but because these are all different elements that increase or decrease your trust in a certain study or its results.
Exactly.
And that goes in line with the guidelines, because guidelines could just highlight these different aspects, and for each some questions that you might be asking in order to basically assess your trust in the study itself.
Yeah.
And I now see it maybe something along the lines as, FAIR principles, because FAIR, I hope everybody here heard about FAIR principles. These are the principles that then have implementation in a certain community, because every community will have something specific, but they should follow the main principles. So maybe it's the same with the tools. So you give some basic principles that then everybody who thinks their specific field or design or whatever requires a specific tool can then follow in order to develop that tool. But these are just like, it's really good to have this discussion, because otherwise I probably wouldn't come up with this. Yeah, because I feel it's very difficult to, because of this broadness of ecology, to go too much into detail, because it's going to be, many questions will be irrelevant to many studies, and then you'll just be skipping them or not able to answer them. So what's the point of 40 plus questions if you cannot answer them or they're not relevant? So good
Or
point.
if it takes too long to
Or if
assess,
it takes
because
too
that's
long.
another, it's a barrier. So you need something like rule of thumb, it's like maximum 15 minutes per study, ideally 10 minutes to assess. So,
Mhm.
you know, just looking at the key things
Yeah, yeah.
to see whether they are there or not, that takes around 10 minutes.
Excellent, excellent points. Anybody else would like to add on this?
This point about the time, I
Mm.
see that, but didn't you say that in medicine they spent 240 minutes? Wasn't it? I mean, I still think it's a huge amount. It's 2 hours, but if they are doing 2 hours, how can we say that we only want to do 10 minutes? Like, how do we justify that?
I've read of a median number of studies in a systematic Cochrane. It's like 4 or 5. Yeah, so they can afford to do that for few studies that are included in their synthesis. If we have 100 or 200 studies, we cannot spend that, 2 hours per study.
I'm
And
in agreement
ideally,
with
we
Gosha.
should be able to use AI to help with the assessment as well.
Mm. At one point, but again, this depends on reporting quality.
Yeah,
Which,
so it's really like focus on whether it's there or not, like given element. That kind of indices of trust.
yeah. Yes. Anybody else wants to add something? Okay, if not, I'm really happy with this session. It wasn't many people, but I think it was really useful discussion. Sometimes it's better if it's less because then you get a better interaction and more people actually say what they think. So Google Doc is open, so you can just add any additional things, thoughts that you have to it after the session. Also, at the end of the Google Docs, you have a link, and I'll post it here too. If you would like to stay in touch, well, I don't know, I can't copy paste it for some reason. It just doesn't allow me. But you see it in the Google Docs. Something is with the chat, it doesn't work for me at this moment. Because if you would like to stay in touch, if we decide, we hope we will, but to work on, maybe now it's going to be a guideline or maybe it's going to be something else, but I believe it's going to happen at one point. So if you would like to be part of that, then please leave your contact in the Google form. You can also just email me. And once I managed to copy paste it. Oh, somebody else also ran it. Thank you, Ran. Then we'll just get in touch and see if you would like to participate in this project. But it might happen soon or maybe in 2-3 years. Who knows? It's difficult to predict. Anyway, thank you a lot. This was useful. I also have a look at your answers during the hackathon. And I think with that, we can close in time our session. So you can go to the next one. Thank you.
Thank you.
Ciao, ciao.
Bye.
Thank you. Bye.
Thank you. Bye.